Welcome, friend. Welcome to our channel Quality Guru. Friend, today our topic is good laboratory practices in pharmaceutical industry. GLP originated in the 1970s from the US Food and Drug Administration in response to widespread fraud and misconduct in toxicology lab that submitted falsified safety data for drug and cosmetic products. The FDA's 1976 proposed rules lead to their 1976 final rule 21 CFR 58, which provided a framework to ensure the quality and the integrity of non-clinical safety study. These principles were subsequently adopted and expanded upon by the Organization for Economic Cooperation and Development, that is OECD, in the year of 1981, creating international GLP standards that are now fundamental to pharmaceutical safety assessment. Precursor and catalyst. In 1970s, cases of fraud. The foundation of GLP was led by major scandals involving toxicology laboratories such as Industrial Biotest Lab, that is IBT, that falsifies safety data submitted to the FDA for pharmaceutical and cosmetic companies. FDA response. These incidents led to public scrutiny and resulted in the FDA proposing regulation to ensure the accuracy and the integrity of data submitted to them. FDA establishment of GLP late 1970s. 1976 proposed rule. The FDA first outlined GLP principle in proposed regulation in 1976. 1979 final rule. The final FDA GLP regulation 21 CFR 58 was established in 1979, setting a framework for the planning, performance, monitoring, recording, archiving, and reporting of non-clinical regulatory studies to support application for FDA regulated products. International adoption by OECD in 1980s. 1981 OECD principle. The Organization for Economic Cooperation and Development, that is OECD, established international principle of GLP in 1981 based on the FDA groundwork. Global impact. The OECD principle promoted the acceptance of safety data that across member countries, facilitating international trade and the development of globally regulated products like pharmaceuticals. Evaluation and modification. 1995-1996 revision. A team of experts revised and modified the OECD principle of GLP and in the year of 1995 and the year of 1996, leading to the current consensus-based revision. Ongoing compliance. The principle continued to be a cornerstone of the pharmaceutical industry, ensuring that quality and integrity of non-clinical safety data are reliable and can be confidently used for risk and safety assessment by regulatory bodies worldwide. Good laboratory practices is a quality system of guidelines and principles for organizing, performing, monitoring, recording, and archiving health and environmental safety study. The goal of GLP is to ensure quality, consistency, reliability, and the validity of laboratory data, which is crucial for regulatory review and approval of product in pharmaceutical industry. Key component includes well-defined study protocol, qualified personnel, proper documentation, validated equipments, adequate facility, and independent quality assurance, that is called QA of QC or laboratory QA. Purpose of GLP: reliability, quality, regulatory compliance, public health, and environment. Reliability: to ensure the data generated is accurate, reliable, and reproducible. Quality: to maintain a high standard of quality in the planning, performance, and reporting of quality control. Operation activity. Regulatory compliance: to provide a framework for data that can be submitted to regulatory agency for approval of new products, public health and environment, to support the safety assessment of substances and products for human health and environmental protection. Key principle of GLP: organization and personnel, facility and equipment, study protocol, documentation and record keeping, quality assurance, archiving. Organization and personnel, clear defined roles and responsibility, that is job responsibility, and proper training file for all QC personnel, and qualification of QC analyst, that is called analyst qualification. Facility and equipment, appropriately designed and maintained facility along with calibrated and well kept equipment. Study protocol, standardized and well defined detailed protocol for planning and execution of study. Documentation and record upkeeping, comprehensive and accurate record of all study activity, data and result. Quality assurance, an independent quality assurance unit that monitor compliance with GLP, that is called QA of QC or laboratory QA. Archiving, proper storage and retrieval of records and data in a secure archive. Key component of good laboratory practices: personnel and training, qualification and training. All personnel, that is QC analyst, must possess the necessary qualification, that is analyst qualification, and receive adequate training for their assigned task, that is job responsibility. Defined job responsibility, roles and defined roles. A clear organizational structure should define roles and responsibility for laboratory personnel, that is define organogram and define job responsibility for each and every QC analyst. Personal protective equipment, that is PPEs. Appropriate PPEs must be used at all the time. Facility and equipment, design and maintenance. Facility must be appropriately designed, maintained and kept clean to prevent interference with the study. Calibration and maintenance. Equipment must be properly calibrated, maintained and have written operating and maintenance procedure, that is defined SOPs. Segregation. Equipment and study areas should be segregated to prevent cross contamination. Study planning and management protocols. Study must be conducted according to detailed written protocols. Quality assurance. A dedicated QA unit independently monitor study to ensure adherence to GLP principle. Archiving. All study related record and material must be properly archived and retained. Documentation and record upkeeping. Standard operating procedure, that is SOPs. Written SOP must be available for all routine procedure. Traceability. All activity data, OS, OT, laboratory incident and deviation must be clearly documented to ensure traceability and reliability in the QC operation activity. Data integrity. Record must be accurate, complete and maintained for the required retention period. Quality control. Method validation. Analytical method must be validated to ensure their accuracy and reliability. Reagent control. Reagent and sample must be properly labeled, stored and documented. All reference standard, working standard must be procured from approved vendor, and that is from IPRS, and it should be stored in a defined 2 to 8 degrees storage condition. Audit. Regular internal and external audit ensure that the quality system is effectively implemented. Why GLP is important in the pharmaceutical industry? Data reliability, regulatory acceptance, product safety, scientific integrity, data reliability. GLP ensures that the data from non-clinical safety studies is robust and reliable, which is essential for making informed decisions about drug safety and efficacy. Regulatory acceptance. Study conducted under GLP are accepted by regulatory bodies worldwide, reducing the need for repetitive study and facilitating the approval process for new drugs. Product safety. Ultimately, GLP contribute to the development of safer and more effective pharmaceutical products, protecting consumers and the environment. Scientific integrity. GLP uphold the integrity of scientific research and builds confidence in the result generated by pharmaceutical laboratories. Key benefits of GLP. The key benefits of good laboratory practices in the pharmaceutical industry include data integrity and data reliability, regulatory compliance, accelerated drug development. Data integrity and data reliability. GLP mandate meticulous record upkeeping, well-defined procedures and rigorous quality control, ensuring the accuracy, traceability and the reproducibility of data from non-clinical studies. Regulatory compliance. Adhering to GLP is a mandatory requirement for conducting non-clinical studies that support drug application, helping companies meet regulatory standards set by agencies like the FDA's. Accelerated drug development. Reliable, high-quality data from the outset prevents study failures and reduces the need for additional testing, which streamlines the drug development process and speed time to market. Public health and safety. By providing sound, validated data on a drug safety and efficacy, GLP helps protect human and animal health by ensuring that only safe products reach human clinical trial and the market. Global data acceptance. The international harmonization of GLP principle allowed test data generated in one country to be accepted by a regulatory agency in other, reducing research duplication and facilitating global collaboration. Enhanced reputation and trust. Compliance with GLP demonstrate a commitment to quality and ethical standard, building confidence and trust with regulatory authority, investors and the public. Reduce risk and cost. GLP's structured environment and emphasizes on error prevention, minimize the risk of data manipulation, fraud, and inconsistency, which can lead to costly delay, rejection, or setback in the drug development program. Key takeaway for GLP. The key takeaway for good laboratory practices in the pharmaceutical industry is that it ensures the data integrity, data reliability, and the reproducibility of non-clinical studies, thereby
preventing duplication of research and fostering public trust in pharmaceutical products. GLP achieves this by establishing a robust framework of procedures for facility, equipment, personnel, and data handling to produce accurate and credible results for drug safety and efficacy. Thanks a lot, and keep watching our channel Quality Guru. It's a small initiative and knowledge sharing platform for our pharmaceutical professional. Like, comment and subscribe please more and more. Thank you.